Hello, my name is Viktor Stolc. I'm a senior scientist at the NASA Ames Research Center. Thank you for the invitation to present our research results on the effects of reactive oxygen species in mice flown on the International Space Station and how excess reactive oxygen species drives mitochondrial dysfunction and health risks, especially in the brain. As I'm sure most of you will recall, the brain consumes more oxygen, which is the source of reactive oxygen species, than any other tissue in the body. This presentation is based on a long-standing collaboration with colleagues from NASA, Stanford, SETI, and the Slovak Academy of Sciences. An overarching theme for our effort is the topic of timekeeping in biological systems. We use genome science technology, high-performance computing, and next-generation sequencing to do our work. I'll start with a little cartoon of myself just to make the point that if you remember nothing else from this talk, I would like you to take with you the understanding that reactive oxygen species can be both good and bad, and yes, even ugly when it comes to human physiology, especially the physiology of the brain. My apologies to Clint Eastwood. If any of you in the audience are movie buffs, you may recognize that the phrase "make my day" is not from the movie The Good, Bad, and the Ugly, but I thought I would combine these phrases because they make so much sense for my talk. Brain development depends on reactive oxygen species. For example, reactive oxygen species trigger neurogenesis, neural differentiation, and neurite outgrowth, a process that is dependent on NADPH oxidase, or NOx, with several isoforms. NOx2 knockout mice exhibit a reduction in the level of hippocampal neuronal progenitors in the adult brain, suggesting that basal reactive oxygen species levels are instrumental in maintaining neurogenic niches. Normal brain function also depends on reactive oxygen species. Physiological levels of reactive oxygen species support crucial cellular processes in the brain, acting as second messengers and regulating intrinsic signaling pathways. Reactive oxygen species also play an important role in regulating synaptic plasticity and memory, with measurable effects on long-term potentiation in the hippocampus. This is ROS acting in a good way. In contrast, virtually all diseases of the brain involve perturbation of the normal levels of oxygen species. For example, glutamatergic excitotoxicity due to sustained glutamate stimulation includes permanent activation of NOx2 in hippocampal neurons, leading to neural degeneration. Reactive oxygen species imbalance occurs in all known degenerative neurological disorders, including Parkinson's disease, Huntington's disease, and infectious encephalitis. Alzheimer's disease and other forms of dementia are also accompanied by significant alterations in reactive oxygen species levels. Superoxide anion, O2-, hydroxyl radical, OH-, and hydrogen peroxide, H2O2, all play a role in neuronal degeneration and other brain diseases. This leads to a question: might systemically elevated ROS contribute to brain disease in the same way that reactive oxygen species generated locally in the brain can cause central nervous system pathology? To answer this question, it is helpful to take stock of where we stand with the epidemic of metabolic syndrome or metabolic dysfunction that we face in the US and even worldwide. The problem of metabolic dysfunction is truly enormous. It is a huge problem in the US, but it is a problem that extends globally. Despite the use of obesity drugs and diabetic agents, the incidence and prevalence of metabolic dysfunction is in the hundreds of millions, annually increasing at an inflation rate of greater than 10%. This clearly is a tremendous burden on the healthcare costs in the US, totaling $3.2 trillion per year, and as much as 75% of this cost is attributable to chronic metabolic diseases. So this is clearly not a problem of genetics. This is a problem of overeating and metabolism. The good news is that it is reversible. So metabolic dysfunction or syndrome includes the following diseases: cardiovascular disease, hypertension, diabetes, lipid abnormalities, non-alcoholic fatty liver disease, cancer, dementia, and even sleep disorders. Additionally, clinically recognized subcellular pathologies associated with metabolic function include oxidative stress, where excess reactive oxygen species are involved, glycation of proteins affecting all vasculature in the body, especially the microvasculature, mitochondrial dysfunction, insulin resistance, inflammation, epigenetics, including methylation of DNA, and DNA mutations, as was revealed by genome-wide association studies. So the question arises, what do these cellular dysfunctions have in common? We now know that it is excess reactive oxygen species. So what is happening in the brain? Well, high carbohydrate intake and other aspects of metabolic dysfunction are linked to changes in brain function in areas governing appetite and reward addiction, especially in the hypothalamus. Several studies connect insulin resistance and metabolic issues to neurological conditions like Alzheimer's disease. Emerging evidence indicates metabolic dysfunction in neurodegenerative diseases is a problem too. Too much carbohydrate intake leads to higher inflammation and oxidative stress, impairing mitochondrial function, including mitochondria in the brain. This is an energy problem, paradoxically, where excess carbohydrate intake leads to metabolic dysfunction, paradoxically lowering ATP, the energy of metabolism. So metabolic syndrome diseases like cardiovascular disease, hypertension, diabetes, lipid abnormalities, and sleep disorders are also manifesting in astronauts in spaceflight, where the spaceflight risks of excess reactive oxygen species affect their health. So I'm sure that everyone in the audience will appreciate that sleep is critical for human health and is required by all mammals. Therefore, it is natural to ask, what is the origin of biological timekeeping on Earth and in orbit on the International Space Station? How is biological timekeeping altered in space in humans and organisms that are used to study circadian rhythms on Earth? Timing and duration of sleep are controlled by the circadian rhythm, which keeps a 24-hour internal rhythm that synchronizes to environmental stimuli. All organisms from mammals to prokaryotes have an internal clock adapted to the Earth's rotational schedule of 24 hours. However, by contrast, the International Space Station orbits within 93 minutes, creating a 93-minute day-night cycle with different environmental stimuli. And reactive oxygen species are involved in disordered sleep physiology from a variety of causes, including spaceflight. Reactive oxygen species serve a timekeeping function as switches of various cellular processes and physiological rhythms. The best candidates for timekeeping functions are reactive oxygen species oscillations, such as in hydroxyl radicals, which are produced as byproducts of bioenergetic metabolism. A complex network of coupled multiple feedback oscillators, such as temporally controlled RNA transcription and periodic proteins and enzyme activities, are coordinated by the redox cycle. These result in various biological phenotypes such as proliferation, differentiation, autophagy, immune functions, and aging. To counteract excess reactive oxygen species, cells contain antioxidant systems, including para-redoxin enzymes, which protect against oxidative stress. These para-redoxin enzymes, in essence, serve as markers for circadian rhythms and circadian systems. Mutants with defective para-redoxin enzymes exhibit increased somatic mutations caused by excess reactive oxygen species. More about hypermutation as a consequence of gene activity during the oxidative phase of the redox cycle is on the next slide. Studies in yeast provide a wonderful example of how the redox system is cyclical. Even though yeast don't have lungs, they consume oxygen in a cyclical manner, as shown in the top left panel, much like breathing. Redox cycle generates an oscillating pattern of gene expressions also, as shown on the bottom left panel. Another way to say this is that some genes are expressed in the oxidative phase and different genes are expressed in the reductive phase. The right graph shows that genes transcribed at the oxidative phase accumulate mutations significantly more than genes transcribed in the reductive phase, and the mutation frequency is plotted on the y-axis. Therefore, environmental stresses that induce reactive oxygen species, such as in spaceflight, have the potential to mutate DNA and disrupt biorhythms simultaneously. One of the most interesting points about this data is that reactive oxygen species induced by the environment enable a coupling between heredity and the environment. This is why studying life processes in space provides a window of opportunity to unravel the timekeeping functions and really all cellular functions at a fundamental level. Based on the timekeeping functions of reactive oxygen species oscillations in yeast, we explored the biochemical effect of the redox cycle in mice aboard the International Space Station to determine if environmental stress affected their gene sequences and circadian rhythm. A special habitat is used on the International Space Station called the Rodent Research Hardware System Platform, which was designed at NASA Ames, as shown on the right panel. Here is a movie of mice activity on the International Space Station. This is an environment for the mice, but they still exhibit some range of normal behavior, such as self-grooming, locomotion by using their momentum, and grabbing onto the cage to move around. The environment that they are in makes it very difficult for the animals to sleep, even though they appear to be well adapted to microgravity. This becomes apparent after about 14 days on board the ISS, as mice become sleepless and hyperactive. So we can use a simple graph to quantify the activity of mice, and as you can see, they sleep very little on the International Space Station. Most importantly, there is a dramatic increase in the activity of mice in space on the ISS in comparison to ground control mice. Quantitative behavior or activity assay revealed that mice became sleepless in about 14 days on the International Space Station. Now let's turn our attention to another consequence of excess reactive oxygen species, namely somatic mutation, which is shown on the next slide. So for this experiment, 16-week-old mice were sent to the International Space Station with equal number of control mice kept in nearly identical rodent habitats on the ground at the Kennedy Space Center. To determine the rate of somatic mutation in spaceflight, RNA sequences from different tissues of mice were determined by next-generation sequencing. The web links for the NASA spaceflight samples and data collections for these experiments are shown at the bottom. Somatic mutations were determined by a novel analytical pipeline that was developed at NASA Ames Research Center. If nothing unusual were going on, we would have expected 1 or 2 somatic mutations during the period of the experiment, which lasted only 40 days on the International Space Station. In fact, we observed many thousands of mutations in each tissue that was examined. By comparison, essentially no somatic mutations were detected in the ground control mice. As shown in the Venn diagram, some somatic mutations were unique to specific tissues, some were shared, and several of the mutations identified were common to all 4 types of tissues that were analyzed. These are the hotspots of variation in the mouse genome. The International Space Station induced mutations that lead to low level of genetic mosaicism in different tissues, and some of the non-synonymous mutations, those that change the amino acid identities in encoded proteins, likely resulted in the loss of function in areas of physiology relevant to spaceflight risks. In comparison to the known mutations that accumulate over the lifespan of an organism, the International Space Station induced mutation rate is up to 3 orders of magnitude higher than on Earth in every tissue examined in just 40 days. This is quite an amazing result. Additionally, the genes that are expressed the most exhibited the highest rate of somatic mutation. So this raises a question: are reactive oxygen species induced somatic mutations completely random? Are A's, T's, C's, and G's being altered with the same frequency? Well, the simple answer is no. It turns out that guanines are substituted more frequently, and the data are shown on the next slide. We know from chemistry that guanine is the most easily oxidized of the nucleotide bases, a process that can occur because of endogenous production of reactive oxygen species. And out of the 12 possible base substitutions, the preference for guanine substitutions was observed in all but liver tissue types, which is consistent with the notion that elevated reactive oxygen species generation on the International Space Station led to the preferential oxidation of guanine and therefore its substitution. So, in summary, reactive oxygen species play a crucial role in the central nervous system in both normal development and adult physiology, and play a critical role in pathophysiology as well. The current epidemic of metabolic dysfunction that is now seen worldwide forces us to think about an even broader role of reactive oxygen species in pathophysiology throughout the body. The range of pathologies that NASA is concerned about for astronauts bears a striking resemblance to the range of pathologies caused by metabolic dysfunction, indicating that reactive oxygen species is the likely common link. The heightened oxidative stress from metabolic dysfunction may contribute to mutations and cellular damage, especially in the central nervous system, via mechanisms that link spaceflight physiology with terrestrial pathophysiology. So with this, I conclude the presentation and thank you for your attention.